that the junction between the two neurons, it could be presynaptic neuron and postsynaptic neuron, meaning it is a junction present between the two neurons through which information can be passed from one neuron to the another neuron. That gap between the two neurons, you can call it as a synapse. So here, synaptic transmission. Synaptic transmission, nothing but intracellular communication within the nervous system. What is meant by intracellular and intercellular? Intra and inter.
Inter means between the cells, intra means in the cell.
Yeah, inter means between the cells, intra means within the cell. So here we have a intracellular communication, means between the cells. The cells will communicate from one cell will communicate the neighboring cell through the transmission or through any message, that we can call it as a intercellular communication. This will take place in the nervous system. So now it is carried out through the junctional contacts, or that we can call it as a synapse. Some synapses involve the close opposition of the two cell membrane. Ionic channels lined up so the direct electrical communication takes place. How information can be passed in the one neuron to another neuron? Anybody will send a message to one neuron to another neuron? Then what kind of information will be propagated between the cells or neurons? How it could be the new information? Is it in the form of channels or electrical signals or chemical signals?
Ion channels.
Yes, ions will help to communicate from one neuron to another neuron. What kind of ions involved during the communication? What kind of
Na and K.
Calcium.
Calcium and
calcium?
Potassium also.
Sodium, potassium, calcium ions are involved during the cell communications. Okay. Here we have the signals will be in the form of electrical communication takes place in the cells. The synapse transmits signals across a gap between two cell membranes with the aid of chemical transmitters and are therefore called chemical synapse. If the signal will pass from one neuron to another neuron in the form of chemicals, that we can call it as a chemical synapse. Then here we have a presynaptic neuron resides in a close opposition to the postsynaptic neuron. So you will have a diagram in your textbook. One is a presynaptic neuron, then another one is a postsynaptic neuron. This presynaptic neuron and postsynaptic neuron are separated by a gap or a space between the presynaptic neuron and postsynaptic neuron. That gap, you can call it as synaptic cleft. What is it meant by synaptic cleft?
The gap between the presynaptic neuron and the postsynaptic neuron.
Yes, the gap or the space present between the presynaptic neuron and postsynaptic neuron, that gap you can call it as a synaptic cleft. The terminal of the presynaptic area synthesizes a neurotransmitters. Where neurotransmitters are synthesized? Terminal portion of presynaptic area. Mostly neurotransmitters are synthesized and stored in the presynaptic neuron area only. From this presynaptic area, this neurotransmitter will transmit from presynaptic to postsynaptic neuron. What is meant by neurotransmitters? Anybody have an idea? Can you tell me any of the neurotransmitters? Any one of the neurotransmitters?
Chemical substances.
Yes, the chemical substances. Can you name any one of the neurotransmitters?
Acetylcholine.
Acetylcholine, epinephrine, norepinephrine, dopamine, serotonin, These are all kind of neurotransmitters. Mostly neurotransmitters are synthesized in a presynaptic terminal only. And where they are stored, these neurotransmitters and stores its membrane vesicles. In the space of presynaptic neuron, there is some vesicles present in the presynaptic neuron. In these vesicles, these neurotransmitters are stored. Where neurotransmitters are stored, whether in presynaptic neuron or postsynaptic neuron.
Presynaptic.
Presynaptic neuron.
In the presynaptic neuron, there will be a vessels. In that vessels, these neurotransmitters are stored. So the presynaptic terminal is triggered through an influx of calcium ions by an action potential. The vesicle contents are released through exocytosis of the neurotransmitter pockets into a synaptic cleft. The neurotransmitter molecules diffuse across the cleft and bind either to catabolic enzyme, where they are inactivated, or to postsynaptic receptors. The terminal of the postsynaptic cell detect the presence of transmitter and initiate a postsynaptic response, either in electrical changes in the permeable to ions or a metabolic changes in the activity of intracellular enzyme coupled to the receptor. So whenever the presynaptic neuron releases the neurotransmitter, for example, epinephrine neurotransmitter is released from the presynaptic neuron. So how this presynaptic neuron will release the neurotransmitters? How? When will be released the neurotransmitter from presynaptic neuron to postsynaptic neuron? By which action? The presynaptic neuron is triggered through influx of calcium ions. Whenever the calcium ions is triggered out due to action potential. What do you mean by action potential? What do you mean by action potential? Anybody have an idea? Have you heard about action potential? When action potential is generated in the now?
When we give any response.
What, Amma? Action potential is nothing but it generates the nerve impulse whenever the charge could be plus 80 millivolts above the threshold value or up to the threshold value. This action potential can be generated. Whenever the action potential can be generated, these neurotransmitters will transform from the presynaptic neuron to the postsynaptic neuron by the action or influx of calcium ions, and the content of the vesicles will be released. In the content of the vesicles, neurotransmitters are present. These neurotransmitters will be released from the presynaptic neuron by the action of exocytosis. What is meant by exocytosis? Breakdown of the cell content, then releasing of neurotransmitters of the vesicles, which is called exocytosis. These neurotransmitter pockets will go into the synaptic cleft. Synaptic cleft, the neurotransmitters will go to the synaptic cleft. From the synaptic cleft, the neurotransmitter molecules are diffused across the cleft and bind to the postsynaptic neuron. That postsynaptic neuron, it could be catabolic, either catabolic enzyme activity can be initiated, or the postsynaptic receptor. The postsynaptic cells detect the presence of neurotransmitters response and initiate the action. It could be electrical action or it could be chemical action. So that is all about the presynaptic neuron and postsynaptic neuron. Between the presynaptic neuron and postsynaptic neuron, there will be the gap is present. That gap you can call it a synaptic cleft. So this presynaptic neuron, some neurotransmitters are synthesized in the presynaptic neuron in the form of vesicles. Whenever these vesicles undergo bursting of the vesicles and release the neurotransmitters, that the action we can call it as a exocytosis. These neurotransmitters bind to the postsynaptic neuron and initiate the action in the postsynaptic neuron. It could be chemical action or electrical actions that will be takes place in postsynaptic neuron. Some receptors may trigger a change in the activity of intracellular enzyme. So this neurotransmitter will alter the action or change the activity in the intercellular enzyme, which is allosterically coupled to the receptor, thereby causing a metabolic responses. So this all kind of metabolic responses will take place in postsynaptic neuron by the action of the neurotransmitters which are released by the presynaptic neuron. Then you have a presynaptic mechanisms. So how it will act in the presynaptic? There will be a mechanism. So they have taken an example of acetylcholine synthesis. So acetylcholine is present at many synapses, most in the vertebrates and invertebrates. This acetylcholine is present mostly in the vertebrates and invertebrates. Acetylcholine is a common neurotransmitter. It is a common neurotransmitter which is present mostly in vertebrates and invertebrates. At vertebrate, neuromuscular junction. What is meant by neuromuscular junction? The junction between the neuron and muscle, which is called a neuromuscular junction. In both the insects and mammals, it is synthesized from acetate and acetylcholine by choline transferase. That is called CAT. CAT is nothing but choline acetyl transferase. So it is synthesized in acetyl-CoA, which is relatively small enzyme and it is located in the cytoplasm of the presynaptic nerve terminals. This acetylcholine is the one kind of a common neurotransmitter which is present in both vertebrates and invertebrates. Mostly this acetylcholine is synthesized in the cytoplasm of the presynaptic nerve terminals. This acetylcholinesterase binds to the enzyme first. Then catalysis occurs when choline binds to releasing a acetylcholine. So subsequently leaves the regenerative to the free enzyme. There will be some kind of chemical reactions take place in the acetylcholine synthesis. There is an equation. So this choline is taken up by an active transport mechanism from extracellular fluids in a mammal. Acetylcholine synthesis can block by the hemicholinum, and 3-hemicholinum is not transported across the cell membrane and does not inhibit the CAT activity, but inhibit the acetylcholine synthesis by blocking the entry of choline to synthesized sites. A similar uptake mechanism exists in insects. This acetylcholine synthesis in cockroaches and subsequent depletion of the presynaptic source of acetylcholine results from the presynaptic stimulation in the presence of hemicholinum. So this is all about the presynaptic mechanism of acetylcholine synthesis. So there will be a metabolism of acetylcholine. Acetylcholine is stored in a synaptic vesicle which is released into the synaptic cleft by action of exocytosis. Acetylcholine molecule diffuse across the cleft and bind either to catabolic enzyme to get the inactivated or postsynaptic receptors. The hydrolysis of acetylcholine is by the action of acetylcholinesterase. The enzyme is polypeptide and has the two active subsites, the anionic site and esteretic site. It has the two subsites, that is anionic site and esteretic site. The anionic site interact with a quaternary nitrogen group. It will be some chemistry. So here the anionic site is always interact with a quaternary nitrogen group, and the esteretic site around serine residues with an acetate group. This will be interacted. The hydroxyl group is rendered more reactive by hydrogen bonding to the imidazole group of histidine residue. The substrate bind to the enzyme, then serinium attack the carbonyl group. of the ester liberating the choline and becomes a acylated. So here some acetylcholine will be altered due to activity of metabolism, due to activity of chemical reactions takes place in the neuron. Acetylcholine is then hydrolyzed state and OH group is regenerated again. The organophosphate and carbamidase insecticide interact with an acetylcholinesterase in the same way the acetylcholine does phosphorylating or carbonylating the serine hydroxy. However, the rate of regeneration of the active enzyme from the complex is higher from the acyl complex than carbonylated or phosphorylated activity of acetylcholinesterase. Dephosphorylation is very slow and may take even months. So much of this inhibition or organophosphate is considered an irreversible reaction. So here there's some regenerated acetylcholine, intermediate acetylcholine, acylated acetylcholine, acetic acid. This all will be in the diagrammatic representation. So in the form of metabolic activity, some chemical exchanges will takes place. This is the interaction of acetylcholine with acetylcholinesterase enzyme activity. That is called metabolism of acetylcholine. Then insecticide and cholinesterase. Insecticides. So though cat can be potential target for new insecticides, cat is nothing but cholinesterase. This cholinesterase acts as a potential target for new insecticides. So far, no commercial compounds have been developed to act. So for the outside head, cat is a 2-isothiocyanidethyl and methylamine iodide. It blocks the access of both choline and acetyl-CoA to the enzyme, but it is not insecticidal. Probably because of higher water solubility afforded by a quarantized nitrogen. A series of amylase and SSCL3 substitute hydronation of inhibit the cat and block the transmission in the cervical nerve signal fiber synapse to the cockroach with altering the sensitivity of postsynaptic membrane to acetylcholine or nicotine. These compounds which are active as an enzyme inhibitors blocked the synaptic transmission while those which were inactive did not. Several S-phenyl and altroacetates were more potent as a cat inhibitors, but the mode of action of these compounds on the intact of synapse is complicated by an antagonism of the postsynaptic acetylcholine receptor. None of the malimides and substituted by hydration, which are active in vitro and insecticidal, even though acetylcholine synthesis appeared to be inhibited. The story of insecticides and cholinesterase, that is called cat. This cat will This cat will not act as an insecticides, but it block the communication system from presynaptic neuron to the postsynaptic neuron signaling pathways. So in that way, this cat can be act as a insecticides. Then neurotransmitters release. How neurotransmitters can be released from the presynaptic neuron? Let us see. Neurotransmitters release. The release of neurotransmitter into the synaptic cleft is accomplished by exocytosis. Exocytosis means the breakdown of the cell content and releasing of neurotransmitters, which is called exocytosis. This exocytosis can be takes place in the presynaptic neuron terminal. When the membrane surrounding the synaptic vesicles fuse with a cell membrane, spilling the content into the cleft. So whatever the content which is present in the vesicle, that will go to the synaptic cleft. During a single episode of release, only a small fraction of the vesicle fuse with the membrane. So all the content of the neurotransmitters will be released from the vesicle, but only a few vesicles or fraction of vesicles fuse with the membrane of the postsynaptic membrane. One row of vesicles are situated less than 50 angstrom from the cell membrane along a region called the active zone. This may represent a radially releasable store of transmitter of non-random position of the vesicle along the active zone, suggests some recognition process, but none has been demonstrated. Vesicle could diffuse one vesicle diameter in about 200, since the upper frequency of nerve terminal stimulation is about 100 Hz. Simple diffusion may account for this. So what happened exactly? Exocytosis is triggered by a calcium influx. Whenever the calcium ions comes into the presynaptic neuron, this will trigger the vesicle molecule. The vesicle molecule which is present in the presynaptic neuron. Due to the triggering action or action potential of the calcium influx into the presynaptic neurons, then exocytosis action will take place. After completion of exocytosis action, it releases the neurotransmitters through voltage-dependent calcium channels. Without calcium influx ions, there will be no exocytosis takes place. If there will be no exocytosis takes place, there will be no neurotransmitters will release. So here the neurotransmitters will be released by the action of calcium influx. By the action of calcium influx only, these neurotransmitters are released. So then dependent calcium channels which open when action potential inverts the terminal in addition of release of calcium ions from internal binding sites. About 80% of delay between the action potential inversion and neurotransmitter release is due to the activation of the calcium channels, leaving only about 2000 angstrom for the calcium to stimulate the exocytosis. Calcium can diffuse only about 2000 angstrom in this 200 is, therefore the calcium channels must be very close to or in the active zone. If one assumes that calcium is evenly distributed in a 2000 angstrom bond A band and next to the membrane of the calcium concentration would reach about 10,000 m. So here the folding increasing of over the resting internal calcium concentration. The quantitative relation between the calcium concentration inside and transmitter release is not known, but the dependence of the release of calcium influx or current is approximately linear. But here the influx of calcium ions exocytosis takes place. By the exocytosis, neurotransmitters can be released. So that is about the releasing of neurotransmitter. How neurotransmitters are released? By the action of influx of calcium ions in the presynaptic nerve terminal. So due to the action of the influx of calcium ions, this vesicle will burst, and by the action of exocytosis, then neurotransmitters will be released. That is the story of neurotransmitters released.
Sir, what is active zone?
What, Amma?
What is active zone?
Active zone means releasing of the neurotransmitters near to the gap junction. That is called active zone. Insecticides and neurotransmitters release. So insecticides like peritonides and DDT analog and 2,2 bis P, ethoxyphenyl and 3,5 dimethyloxetine and EDO causes a massive postsynaptic excitation due to greatly facilitated transmitter to release. A variety of other compounds increase the neurotransmitters release by altering the membrane potential. Sodium channels openers and betaxotoxin and gerantoxin and veratridine, aconitate and other toxins and scorpion, anemone toxins and scorpion toxins increase the influx of sodium into synaptosomal preparations, depolarizing the presynaptic nerve terminals and causing the release of neurotransmitters. All of these compounds broaden the axonal action potential and also depolarize the axon by delaying closing of sodium channels. Compounds that block potassium channels inhibit and delay the increase in potassium current, which determine the falling phase of the action potential. Thus, 4-aminopyridine and broaden that action potential and increase the neurotransmitter release at a synapse. Any compound which broadens the action potential causes a depolarizing upper potential and also cause a repetitive firing and increase the transmitters release. What do you mean by depolarizing? How depolarizing happens in this nows? What is depolarizing? What is repolarizing? What happens during the repolarizing activity?
Channels will be open.
Ion gated channels will be opened. Generally, in an axon membrane, sodium ions are present on the surface of the axon membrane and potassium ions are present inside of the axon membrane. Whenever the action potential generated, the sodium ion gate will be open and sodium ions come inside of the axon membrane. Then the potassium ions goes outside of the axon membrane due to the action of depolarization. So here the depolarization takes place. Insecticide which cause only modest change in the action potential and no change in fingering pattern may cause increase in the transmitters release, which destroy the delicate post-synaptic response. Necessary for meaningful synaptic integration, only fewer sodium channels need to be modified by these compounds to interfere with the synaptic transmission than to interfere with the impulse transmission. So that is all about the insecticides and the neurotransmitters released by the action of the pyrethroids and DDT. Then you have a postsynaptic mechanisms. How postsynaptic mechanism takes place and what are the postsynaptic receptors which are present. A release of neurotransmitter from the synaptic nerve terminal is detected by receptors. So whatever the neurotransmitters which are released from the presynaptic neuron terminal, that will be detected by postsynaptic neurons or postsynaptic receptors bound to the postsynaptic cell membrane. In the case of cholinergic transmission or acetylcholine diffuse across the synaptic cleft. Generally, acetylcholine is released from the presynaptic neuron. From the presynaptic neuron, this acetylcholine is crossed by the synaptic cleft and bind to the postsynaptic receptor. That is called, which will be crossed by the synaptic cleft. Diffuse across the synaptic cleft and bind to the acetylcholine receptors. These receptors are linked directly or allosterically with ionic channels which open in response to receptor activation. And again, depolarization action takes place by postsynaptic neuron. In vertebrates, acetylcholine receptors are classified as a nicotinic or muscarinic based on the ability of nicotine or muscarine to stimulate them. Antagonists are categorized as a nicotinic. For example, tubocrinin, decamethinium, or muscarinic like atropine. Insect nerves contain a pharmacologically distinguishable classes of receptors that are fairly similar but not identical to those in invertebrates. Preparations with high affinity of, for example, high affinity of Kd equals to 1 nm for a bungarotoxin, binding site is a nicotinic, have been found in several insects. In addition to this insect, acetylcholine receptors, which is fit reasonably well into the vertebrate classification system. Insect may have receptors of mixed nature. Thus, a binding of Decamethenism to the receptor from housefly eggs is inhibited by the muscarinic agents. For example, atropine, pilocarpine, and scopolamine, and the nicotinic agents. The nicotine and tuberculinic or binding of nicotine is inhibited by decamethenism and atropine and pilocarpine. This bungarotoxin does not inhibit either nicotine or decamethenin binding. Several peptides have been isolated from the eggs of houseflies, which binds various cholinergic, antagonics, and activity takes place in the postsynaptic neurons or postsynaptic receptors. Some may be bind like a vertebrates, some may not be binding due to the action of yes.
Antagonist means what, sir?
Opposite action. Opposite action, which is called antagonistic activity. Similar action will take place, some will work in a opposite action, which is called antagonistic activity.
Okay, sir.
Then you have insecticides and acetylcholine receptors. What are the acetylcholine receptors? Nicotine bind to the receptor. See, what is meant by receptor? Anybody have idea? Receptor? Receptor.
Receive signals, sir.
Which receive the signal or which receives any information, which is called receptor. Thus, receptor always present on a specific protein. So, for example, here the nicotine bind to the receptor and activate the receptor channel complex. So, whenever the nicotine comes and it is bind with the receptor, then it forms a receptor channel complex. The resulting depolarizes. Whenever it forms the receptor channel complex, then this that cell or that neuron undergo depolarization state. Depolarization state is nothing but the charges will be reversed and it activates the function of neurotransmission, or nerve impulse will be generated by the action of depolarization state. Then it causes the multiple firing of action potential, that spike initiating zone, and behavioral excitation. Whenever the depolarization takes place, then which undergo excitation activity. For example, మనం ఏదైనా పిన్ను తీసుకొని మనం మన బాడీలో ఎక్కడైనా పించ్ చేసినాం అనుకో, గుచ్చినాం అనుకో, అప్పుడు ఏమైపోతుంది, నర్వ్ అనేది అప్పటి వరకు రెస్టింగ్ స్టేట్ లో ఉంటది. ఎప్పుడైతే పెన్సిల్ తో పెన్ తో కానీ మనం మనకు ఎక్కడైనా గుచ్చినాం అనుకో, అప్పుడు మన నర్వ్ అనేది డీపోలరైజేషన్ స్టేట్ లోకి వెళ్తుంది. ఎప్పుడైతే డీపోలరైజేషన్ స్టేట్ లోకి వెళ్తుందో అప్పుడు దాంట్లో ఉన్నటువంటి సోడియం ఛానల్స్ అయానిక్ ఛానల్స్ అన్ని కూడా ఓపెన్ అయిపోయి సోడియం ఛానల్స్ లోపలికి వెళ్ళి పొటాషియం అయాన్స్ బయటికి వచ్చి చార్జెస్ అనేటివి రివర్స్ అవుతాయి. సో అప్పుడు మనం ఆ పెన్ తో కూర్చున్న వెంబడే ఎగ్జైటేషన్ కి గురి అవుతాం. సో దట్ ఈస్ ద ప్రాసెస్ ఆఫ్ ఎగ్జైటేషన్ డ్యూ టు యాక్షన్ ఆఫ్ depolarization. Eventually, the prolonged activation of the channels results in the depolarization of the spike initiating membrane that is sufficiently large to inactivate a substantial fraction of the sodium channels response for the generation of action potential. So whenever, if you want to conduct a nerve impulse, sodium channels are responsible for the nerve impulse, because the sodium channels are present on the outer surface of the axon membrane. So whenever depolarization takes place, the sodium channels get open, and that sodium channels are rushed inside of the axon membrane, and inside there will be a potassium ions. That potassium ions goes out due to reversal action of the, or due to alter action of the ion changes takes place in the axon membrane. So this spike initiating membrane takes place. If you want to generate an action potential, sodium channels are responsible. Without sodium channels, action potential cannot be generated. When this occurs, P channels remain in the resting. So some will be in resting state. activable state to generate a nerve impulse, and conduction will fail. Whenever this sodium potential will be remain in a resting state, there will be no nerve impulse, there will be no conduction of impulse, so impulse conduction would fail due to inactive or due to not activating of the sodium channel. If activating of the sodium channel, nerve impulse and nerve generation takes place. This is the basis of the depolarizing block. Observe this felicitation of synaptic transmission by a variety of drugs. So this action potential and this generation of nerve impulse will be blocked by action of some drugs or blockaging of the depolarizing activity in some kind of insects by spraying of some kind of drugs. So due to spraying of some drugs to the insecticide, this will block the depolarizing activity in the neurons of the insect. So that is all about the, insecticide and acetylcholine receptors. Then we have an insecticides and octopamine receptors. This is also one kind of receptors in the insecticides. So you'll have a farmadin pesticides may activate octopamine receptors. If you use the farmadin pesticide in the crop of an insect, so this will activate the octopamine receptors in the insects. So octopamine, phenylamine, and the catecholamine is a norepinephrine, also act in a similar manner in invertebrates. So this norepinephrine and epinephrine are the catecholamine neurotransmitters, which is same present in the vertebrates like a human being, as well as some the epinephrine and norepinephrine also similar manner function in some kind of invertebrates, mostly arthropods, including arthropods. The amines in arthropods function as both transmitters and neuromodulators, a chemical which modifies the cell response to neurotransmission, which is called a neuromodulator. Neuromodulator are nothing but any chemical which modify the cell response to neurotransmission, which is called neuromodulator. For example, octopamine, which act as a neuromodulator.
Sir, repeat one.
Hello?
Sir, repeat
neuromodulator. Yes, neuromodulator is nothing but any chemical which modify the cell responsibility or cell activity in the action of neurotransmission. Neurotransmission is nothing but transmission of nerve signal from one cell to another cell that will be altered, that will be modified by the action of neuromodulator. Octopamine receptors can be divided into two major categories, with the second category subdivided further into two groups, that is second A and second B. So there will be a farmadin pesticides act as a octopaminic. For example, chlorodamine form causes the excitation of the central nervous system of the tobacco onworm by some action on non-cholinergic synapses. Several biogenic amines, including octopamine, and causes a similar excitation. And then we have another one that is called DCDM, that is desmethyl chlorodiminiform, causes a prolonged glow of the firefly lantern. This glow and that produced by the octopamine are both blocked by phenethylamine and crafropeptimidine, which block the octopamine receptors, but not by pretreatment with risperidone or amphetamine, which causes a depletion of the presynaptic stores of the octopamine. So this octopamine will block the presynaptic stores also by chronic denervation, which causes the degeneration of the presynaptic neurons. Sometimes this octopamine also loses the neurons in the insecticide, that we can call it as a degeneration of presynaptic neurons. Some presynaptic neurons will lose or damaging of the presynaptic neurons. That loss of the neurons or damaging of the neurons, we can call it as a degeneration of the presynaptic neurons. This suggests that an action of DCDM is a postsynaptic rather than a presynaptic. So this is the all activity of octopamine receptors in the insecticides. We'll have a chlorodium aminoform and DCDM induces the release of hyperlipmic hormone from isolated of corpus cardiac of Locusta migratoria. This released by blocked by the androgenic blocker Panaxi-Benjamin, but not by the androgenic blocker propanol by depletion of the presynaptic source of transmitters with the respond. So there are some receptors some receptors are blocked by the action of this neurotransmitter signals. This will act in the some insect, that is called insecticides, and best example for the octopamine receptor. Then we have insecticides and Gaba receptors. So we'll have another receptor which is called Gaba receptor. This Gaba receptor is the most common receptor. So what is meant by Gaba receptors? What is meant by Gaba? Anybody? What is meant by Gaba? Glutamic amino butyric acid receptor. This is one kind of inhibit type of receptor. This macrocyclic lactones isolated from the mycela of streptomyces, which are called avimestin. That is called avium. Avium causes to the nematodes.
What is GABA, sir?
Huh? Hello? GABA is one kind of inhibiting neurotransmitter. Glutamine amino butyric acid. This is a inhibit kind of neurotransmitter or inhibit kind of receptor. So this causes the nematodes to arthropods, causes the paralysis. Paralysis is a kind of diseases to block the signaling transduction in the neurons. So there will be no movement that takes place in the insect, mostly of nematodes and arthropods, through an attack on the GABAergic transmission. This GABA will inhibit the activity of the neurons. Then it causes a disease called paralysis. This paralysis takes place in some kind of insects like nematodes and some kind of arthropods due to the inhibiting activity of the GABA receptors. In both vertebrates and invertebrates, GABA is an inhibitory neurotransmitter. Yes, this GABA is an inhibitory neurotransmitter. Not only in invertebrates, it also inhibits the activity of neurotransmitter in vertebrates also. This is a one kind of inhibitory neurotransmitter that is called GABA. The GABA receptors reside in a postsynaptic membrane and are allosterically coupled to the chlorine channels. Activation of the GABA receptor channel complex increase the membrane permeability to chlorine. This resulting in decrease in membrane resistance, antagonize the effectiveness of excitatory inputs. Generally, any neurotransmitters will transmit the signal from presynaptic neuron to the postsynaptic neuron. So whenever the GABA kind of receptors or GABA kind of inhibitory neurotransmitters, which activate opposite to the other neurotransmitters. Other neurotransmitters are nothing but, we have two kinds of neurotransmitters. One is excitatory neurotransmitters and another one is inhibitory neurotransmitters. The best example of inhibitory neurotransmitter is this GABA receptor. This will act again as the excitatory neurotransmitters. The effectiveness of excitatory inputs by decreasing the arthropod muscle receive both excitatory glutamatergic and inhibitory of GABAergic innervation from the nervous system. So This excitatory neuromuscular transmission by increasing inhibitory of GABA and ergic transmission. This somehow increase the efficacy of GABA-ergic transmission through an action on the postsynaptic. So GABA receptor channel complex that causes the channel activation. Mostly some insecticides bind with the GABA receptors. These GABA receptors are always stop the neural transmitters or nerve conduction, so that whenever stop the neural transmission or nerve conduction, so it leads to communication blocking takes place in the nerve system of the insecticide. Then it causes a disease called paralysis. This mostly takes place in the nematodes and arthropods. These GABA receptors are a kind of inhibitory activity of neurotransmitters or GABA kind of inhibitory receptor. It cannot make the activity of excitation. It stops the activity of the neural transmission. That is called insecticide GABA receptor. Let us see removal of neurotransmitters from the synaptic cleft. Let us see what will happen. Removal of neurotransmitters from the synaptic cleft. Removal of neurotransmitter from the synaptic cleft to terminate the transmission process. What will happen if you remove the neurotransmitter from the synaptic cleft? Anybody? The removal of neurotransmitter from the synaptic cleft to terminate the transmission process. If there will be no neurotransmitter in the presynaptic nerve, there will be no action potential generated. There will be no passage of the neurotransmission from one neuron to the another neuron. This process is accomplished by an enzymatic catalysis by acetylcholinesterase in cholinergic synapses or catechol-methyltransferases and N-acetyltransferase in variety of emergenic synapses, or by active reuptake of neurotransmitters into presynaptic kernel. In the case of emergenic and glutaminergic synapses, only one of this process, hydrolysis of catecholine by acetylcholinesterase, has been implicated in the pesticide action. Cholinesterase are distributed widely in the animal kingdom. They are the hydrolysis which catalyze the breakdown of the cholinesterase to choline. Cholinesterase to choline by the action of catalysis activity and organic acid. Cholinesterase can generally be divided into two main classes. One is Vitro Cholinesterase, also called as pseudocholinesterase, most rapidly hydrolyzed by beta-chlorine to chlorine and butyrate. It shows a little excess substrate inhibition and is found in variety of tissues in mammals, including blood plasma, liver, and nervous system, but not in insects. Acetylcholinesterase is found in erythrocytes, the nervous system and muscle of vertebrates and central nervous system of insects. Acetylcholine is preferred substrate and it exhibits marked excess substrate inhibition. The activity of acetylcholinesterase is to inhibit. Both of these enzymes are inhibited by a organophosphate and carbamate insecticide, but in inhibition of butyroesterase, butyroesterase is not known the function still in the insect. Acetylcholine seems to be distributed widely over the surface of cholinergic neuron and the vertebrate muscles. Mostly, if you remove the neurotransmitter from the synaptic cleft, so which blockage of the nerve impulse cannot be generated, and there will be no depolarization, nothing of the sodium channels to open. This all will be blocked by the removal of neurotransmitter. If there will be a neurotransmitter, the general process will be initiated and action potential will be generated from the one neuron to another neuron. So there will be a chemical equation like a binding site of the acetylcholinesterase and acetylation decarboxylation process. Then insecticides and acetylcholinesterase activity. So here in this, there will be a chemistry involved in this one. So you can go through this so you can understand the insecticide acetylcholinesterase activity. So then physiological consequences of neuron toxication. The poisoning symptoms are produced by insecticides can be easily related to nervous system dysfunction in general, superficial way, to recording the motor output in situ poisoned insect. The definition of death in mammals is relatively straightforward since the prolonged session of breathing or loss of blood pressure becomes irreversible. All of the physiological functions that are associated with the living condition stop, and death, by definition, is inevitable. For practical purpose, then effects of compounds ultimately lead to respiratory or cardiovascular collapse. So mostly toxins are, I told you toxins are two types. One is the neurotoxins and another one is the hemotoxins. Due to neurointoxication, if you take the toxin into the body of any human being or any animal, so thus that toxin can affect in the body, how it can affect in the body, and how it alter or how it changes the normal physiological activity in the body. So that all included in this. So it can changes the blood pressure and some kind of cardiovascular system and some kind of respiratory system will be collapsed by neuronal intoxication. Nothing but neurotoxins. The neurotoxins can be alter the action of normal physiological activity in the body of any insect or human beings. That is called the physiological consequences of neuronal toxification. Process can clear mark insect death. Most investigators considered an insect dead when it is in a moribund and unresponsive to tactile stimulation, but this is not always true. Since this is all about the intoxication of neurotoxification. Once you go through the intoxication of neurotransmission, you will understand some hormonal changes will take place and it disturbs the homeostasis mechanism in the body. So what is meant by homeostasis? Maintaining of equilibrium state in the body that will be disturbed by the action of neurotoxins in the human body or insect body. Then you have a summary about this synaptic transmission. The nervous system collects the information on the environmental condition that uses this information to make a moment-to-moment decision, which determine the insect behavior and internal homeostasis. This information is coded in both electrical and chemical signals, which are compiled, operated on a executed in time-dependent fashion. Relatively small changes in this time dependence by insecticides destroy the delicate interplay between the various signals, which normally result in a coordinating behavior. Generally, nervous system is a kind of a coordinating system. If any channels or if any communications blocked in the nervous system, it will not coordinate the body system. So that is then the excitatory insecticide causes the hyperactivity. Some insecticide causes the hyperactivity in the insect and uncoordinated movements, tremors and convolutions happens some kind of insect. So electrical potential in the nervous system take to form a transmembrane voltages which result from passive diffusion of various permeable ions down their electrochemical gradients. So this is kind of depolarizing activity or repolarizing activity or hyperpolarizing activity. So in this activity, some electrical will be some ions will be diffused inside, some ions will be goes outside. The ability of the nervous system to use the membrane potential to receive the process and send information is based on transient changes in the potential. How nerve transmission takes place? By action generation only the nerve transmission take place or action potential. These changes result from the selective alteration of the membrane permeable to specific ions. For example, specific ions, sodium ions comes inside. what will happen? So depolarization takes place. This is depending upon the which ion entered inside, which ion goes outside of the axon membrane. The information may be coded as a membrane potential amplitude, that is called graded potential, or frequency coded. All are non-action potentials. The ions cross the cell membrane through the other cell member. Any ions can cross the cell membrane, that is called ionic channels. Then we have a variety of synthetic insecticide. So, for example, you have a DDT, perthrives, and the natural toxins interfere with the steady state and transient membrane potential by altering the normal function of the voltage-dependent sodium channels. The compound modify the fraction of the membrane sodium channels in dose-dependent manner. So, and some stabilize the open state. The delay in the channel closing results in prolonged sodium current, which depolarize the membrane for various durations. So depending on the stating kinetic of the channels, modify the various compounds. These compounds which retain sodium channels in open state only transiently tend to producing repetitive firing of action potential. So always sodium channels cannot open. Whenever it is required, then only the sodium channels open, and again it will close. But if it is a prolonged to open the sodium channels, some transient means produce repetitive action potential will take place. While those which retain channels in the open state persistently tend to depolarize membranes. The membrane potential behavior of poisoned neurons and parts of the neurons differ due to special physiological characteristics possessed by a various neuronal membranes to difference in the capable properties of various neurons. The high potency of perthroids is largely attributable to the observation that are relatively few sodium channels must be modified in order to produce toxicologically significant change in the membrane potential. So we'll have an interneuronal communication. Interneuronal means the communication between the neurons, which is called interneuronal communication, is accomplished largely by synaptic connections. So only one neuron will be connected by another neuron by the gap, which is called a synaptic connection between cells. When nerve impulse arouse at a nerve terminal, then it causes influx of calcium ions. So these calcium ions trigger to release the neurotransmitter. These calcium ions are present in the presynaptic neuron, which causes to trigger the neurotransmitter. These neurotransmitters differs across the synaptic cleft. This neurotransmitter will cross the synaptic cleft and bind to the receptor proteins. Across the synaptic cleft and which bind to the receptors of the postsynaptic neuron, some protein changes will be halted. So the protein undergo conformation changes, which either alters the activity of coupled enzymes or any changes in the membrane permeability, or ion changes will be takes place, or chemical changes will be take place, and all the ionic channels will be altered by the action of the neurotransmitters. Ionic selectivity and the channels determine whether given a neurotransmitter will tend to excite, then only it will confirm if it has to perform the activity to excite activity, or it has to perform the inhibitory activity of the particular neuronal transmission. So depending upon the neurotransmitter. If example, for example, one kind of epinephrine is present. Epinephrine is released by the presynaptic neuron. This epinephrine is one kind of excitatory neurotransmitters. Definitely, this will cross with the synaptic cleft and which bind to the receptor of the postsynaptic the postsynaptic. In that postsynaptic, this epinephrine trigger the action of the postsynaptic neuron and it perform the activity of the nerve cell. That is called the positive transmission of the epinephrine. That is called a excitatory neurotransmitter. For example, if GABA neurotransmitter is released from the presynaptic neuron, then it is bound to the synapses. Then it causes the synaptic cleft. Then bind with the receptor of the postsynaptic membrane. Then it inhibits the activity of the postsynaptic neuron. So that is called a kind of excitatory neurotransmitters and inhibitory kind of neurotransmitters. The sum of the postsynaptic potential is decrementally propagated to the spike initiation zone, where it is converted into an action potential. All of the insecticides, for example, we have a more synthetic insecticides that is called DDT and pyrethroids, which influence the shape of the action potential. This insecticide will change and shape of the action potential and frequency of firing, may have an indirect effect on the synaptic transmission. Multiply firing and prolonged depolarization of the presynaptic nerve terminal result in increased neurotransmitter release. Cyclodines and lydin probably have a more direct effect on the neurotransmitter release mechanism, but the molecular details are not yet known. So what happens if you take intoxication of this insecticide like a DDT and pyrethroids? So this DDT and pyrethroids will act on the presynaptic neuron. From the presynaptic neuron, it is release the neurotransmitters. So generally, neurotransmitters released by 1 or 2, depending on the action of the presynaptic vesicle. But if you intoxication of the toxins like insecticides like DDT and pyrethroids, if you take the DDT and pyrethroids DDT and pyrethroids are intox with the insect, it causes to release a more number of neurotransmitters. That is called the activity of synthetic insecticides like a DDT and pyrethroids. Several group of insecticides are interfere with the synaptic transmission. So because of these insecticides, so this communications gaps will be blocked up, and there will be no proper synaptic transmission by the effect of the insecticides like a DDT and pyrethroids. By interaction with the postsynaptic receptor, for example, nicotine or related compounds bind to stimulate the acetylcholine receptor, which depolarize the postsynaptic neuron, leading to excitation. For example, if you take the cigarette, so what will happen? The cigarette will stimulate the acetylcholine receptors and nerve impulse will be generated. So the particular person who is smoking, he will be in a excitatory state for the action of nicotine. That nicotine is present in the tobacco of cigarette. So that is the action takes place. So then depolarizing is nothing but generating the nerve impulse. Depolarizing postsynaptic neuron leading to excitation, eventually synaptic blockade. So then we have a GABA receptor. GABA is the inhibiting activity channels. Then two of the most successful class of insecticides like organophosphate and carbamates interfere with the cholinergic synaptic transmission. So this cholinergic transmission is inhibiting acetylcholinesterase activity. This inhibition is accomplished through the phosphorylation or carbamylation of serine hydroxyl residing in the catalytic site of acetylcholinesterase. So the inhibition of this enzyme result in accumulation of acetylcholine in the synaptic cleft. So this when it is inhibiting the action of the acetylcholinesterase at synaptic cleft, there will be no neurotransmitter takes place in the postsynaptic membrane. So this is all about the how this presynaptic neurotransmitters are released, and whenever it is crossed with the synapses, and it changes or alter the activity of the post-neuronal action. The series of events which are related to nervous system intoxication to insect mortality. So this is mostly takes place in the insect nervous system. If any insect takes the intoxication, means if any insect takes any kind of pesticides or insecticides, it leads to changes in the neuronal activity, and which will inhibit the activity of nerve impulse or nerve conduction or nerve transmission. So then blockaging of this communication in the insect, it leads to death. That is called the insect mortality. Then largely because the collapse of any single physiological, sometimes it also alters the physiological system, and sometimes it also leads to death. So this is all about the neurotransmitters and the insecticides, how it interact with the neurotransmitters and insecticides in the insect. So mostly I have completed half of the portion in the offline classes. That there will be a model questions. Describe an ideal as to how nerve impulse is transmitted in the axon. Already I told in the offline classes how nerve impulse will be transmitted. Nerve impulse etla transmit avutadi, etla generate avutadi, daniki ఏ మిల్లీవోల్ట్ ఎన్ని మిల్లీవోల్ట్స్ ఉంటే జనరేట్ అవుతుంది. దాన్ని అవన్నీ మేము లాస్ట్ క్లాస్ లో we have discussed that. So then now describe how is the acetylcholine synthesized. So just now we have discussed in the story of acetylcholinesterase. So how this acetylcholine is synthesized. Mostly acetylcholine etla synthesize aito dani story rale. It will be asked in a small question. Then what are the different neurotransmitters found in an insect nervous system? So what kind of neurotransmitters which are found in an insect nervous system? Mostly this will be asked in a short type of question. What do you understand by the terms membrane potential, resting potential, and action potential? So this is already we have done in the offline classes. What is meant by membrane potential? What is meant by resting potential? What is meant by action potential? So already we have taken this class in the offline. So already I clear out the resting membrane potential and membrane potential and action potential. So if you go through these 4 questions, so this is the most important questions. You can go through this.
Sir, different neurotransmitters found in insect nerve system means which we have
Edama?
different
Describe?
neurotransmitters found in insect nervous system.
Oh,
From where we have to
different kind of nervous, different neurotransmitters.
found in insect nervous system.
Okay. Cholinesterase, then dopamine, ఇవన్నీ దాని కిందకి వస్తాయమ్మా. Gaba is also one kind of
metabolism కూడా రాయాలా సార్? మెటబాలిజం కూడా రాయాలా సార్?
మెటబాలిజం అవసరం లేదమ్మా, అది జస్ట్ అది షార్ట్ క్వశ్చన్ కింద వస్తుంది కాబట్టి ఓన్లీ ఆ న్యూరోట్రాన్స్మిటర్స్ గురించి రాస్తే అయిపోతుంది. acetylcholine గురించి, గాబా గురించి, లేకపోతే జస్ట్ acetylcholine సింథసిస్ గురించి రాసినా సరిపోతుంది. మెటబాలిజం అవసరం లేదు, సింథసిస్ గురించి రాస్తే సరిపోతుంది.
ఓకే సార్.
2 మార్క్స్ మన షార్ట్ క్వశ్చన్స్ లో వస్తుంది కాబట్టి. సో దీన్ని పీపీటీ చేయాలనుకున్నాం కానీ ఇదంతా కూడా కెమికల్ స్ట్రక్చర్స్ ఉన్నాయి. ఇది పీపీటీ చేసినా మీకు యూజ్ ఉండదు, నాకు కూడా యూజ్ ఉండదు. సో ఇది టాక్సికాలజీ కాబట్టి అంతా కెమికల్ రియాక్షన్స్ జనరల్ గా ఉంటది. సో దీనికి పీపీటీ టైం తీసుకుంటది అనమాట. అది మీకు కూడా పెద్ద యూజ్ఫుల్ ఉండదు ఇదంతా సెటప్ చేయాలనేసి డైరెక్ట్ చెప్తున్నాను. యాక్చువల్లీ ఇది ఆఫ్లైన్ క్లాసెస్ చెప్తేనే మీకు కొంచెం క్లియర్ అవుట్ అవుతుంది. సో ఎనీవే, ఎనీ డౌట్?
Excuse me, sir.
Hello?
Yes, sir. If possible, you can explain in the board itself.
Definitely, I'll try to explain in the board next class. Sure. Definitely, I will explain the board.
Thank you, sir.
Okay. Okay, then I will wind up. Then the next class I will start another chapter. I try to make up in the board. Okay. Thank you all.
Thank you, sir.
Thank you.